Hello, champ. We are continuing with the protein structure, and we learned about amino acid chemistry, how they form secondary structure from the primary structure, alpha helix, beta plated seed, and then uh, various kind of forces that are mainly responsible for protein secondary structure, tertiary structure, okay, different bond, and yeah, Ramachandran plot, that's a very important plot. And today we will learn about protein folding. Okay, trust me or not, that's the really puzzle. Okay, so you will appreciate that how a protein is folding and what is the basic science. That's more than something engineering. Okay, or you can say art. Okay, now just assume that you have a length of something amino acid. Okay, now your amino acid, so you have suppose 10 amino acid or 10 to 100 amino acid, whatever. Just this is N terminal, this is C terminal. Okay, now this is the primary structure. Here from here you make the secondary structure, it's like that. And again that part is forming beta plated seeds, that is a loop, that is beta plated seeds, and after that that's again forming something random coil. But how you define that after secondary structure, how your molecule can be like that, or your molecule can be from here again, then from here one another, then something random coil, like which shape it is going to take. Okay, it can take multiple shapes because there is no such restriction, right? It can fall any way. Suppose if you have some random wire, you can easily see that. Suppose if you take some paper and you can just fold the paper in any pattern, anything like that, also protein can take any uh, suitable folding pattern. Okay, but how it will be decided and why the folding is really important? So the first question is why the folding is important. What do you think, why the folding is important? So first thing, so is the folding depends on something? Yes, the folding depends on something. What is that? We will see later. Second thing is that, is folding responsible for function of protein? Of course. Suppose you have 10 amino acid K, L, A, G, S, P, T, S, A, G, but you can make multiple fold, and with the same amino acid sequence you can generate different scaffold that will perform different function. Maybe it can be a transport protein, maybe it can be some part of hormone, or maybe some stimulating factor, anything, right? So the folding pattern or maybe one enzyme is really important. Suppose in a enzyme you want the active site, and depending on the folding, the active site enzyme will be accessible, so that can convert the substrate to product, right? But if your folding is different, then the active site will not be exposed and your substrate will not never be converted to the product. That's why folding is important. But the question is, folding is dependent on factor, what I mentioned earlier. What the factor is, that is called amino acid sequence or primary structure of protein. So the basic key is the primary structure of a protein. Okay, how it was determined? It was determined by one experiment called Anfinsen's experiment. Anfinsen's experiment. What Anfinsen actually did, we will see. So Anfinsen took ribonuclease. Okay, here's a graphic presentation. So this is ribonuclease. Now, this is native state catalytically active. Now, if you add, okay, urea and mercaptoethanol, what is the role of urea? Urea is a denaturing agent. So, it will disrupt the hydrogen bonding And what is the role of mercaptan ethanol? That is the reducing agent. It is reducing and it's denaturing. Now what will happen? Due to mercaptan ethanol, all the disulfide bond that's present, that will be broken, of course. Now they are converted to sulfhydryl group, and due to denaturation, the protein is in unfolded state. All the hydrogen bond is broken, right? Now here the denaturing agent is urea, and beta mercaptoethanol is also responsible for reducing condition. Okay. Now when he removed the urea and beta mercaptoethanol, he observed that after certain time, okay, maybe it is 8 hours, 10 hours something, but after certain time, the protein is again folded in the same pattern, okay, as it was in native state. How you can visualize it? Suppose in native state it is active. So when you take the urea and beta mercaptoethanol that was removed, actually the protein is again active. You can give the substrate and you can see the conversion to product. So that indicate what? That means whenever you are removing urea and beta mercaptoethanol, your protein started again refolding itself. That means you can do anything with your protein, but how the protein will be folded, that is decided on the sequence of the amino acid. See here, the 40 and 95 they are forming disulfide bond, 5800 they are forming disulfide bond, 70 to 65 they are forming disulfide bond. But why not 40 with 84? Why not 40 with 72? Why not? You have to think it. So that is evolutionary conserved. So remember, protein folding is evolutionary conserved. Evolutionary conserved. Right? Now after refolding also again, you can see there is a bond between 6572, 4095, 110, 58. Like as usual, the disulfide bond were in native state. The same thing happened in the refolding condition. Okay, so another additional experiment was done here. What he did, suppose when he removed beta mercaptoethanol and urea, it took almost 8 hours. But what he did, so he removed urea minus DTT or beta mercaptoethanol, plus he adds some enzyme PDI, protein disulfide isomerase. At that time, the protein folding was much faster. So suppose in that case it's taking 30 minutes to 1 hour. So that means the protein folding is guided by some protein, right? So what information we got from here? The first thing, the protein folding is evolutionary conserved. Second thing, the protein folding is sometimes uh, guided by some enzyme, right? Okay. Now uh, we will see Levinthal paradox. Why the concept of this kind of enzyme like PDI is coming? Levinthal paradox. That is very important concept. From here every times one or two questions is to come. So the Levinthal paradox is saying, so suppose you have protein that consists of n amino acid, or suppose consists of x amino acid. X, that equals to number of amino amino acid, and your protein, each amino acid, each amino acid can take n conformation, n conformation, in different conformation. Then number of conformation will be, that is n to the power x. Now if one protein, one amino acid, if one amino acid take time t time to be in proper place. That means to be properly folded. So n to the power x will take what? N to the power x into t time, right? Now, if I tell you that suppose you have 100 amino acid. Each amino acid will take four different conformation. Four different conformation. Now, what will be the total number of conformation? Total number of conformation, that is 4 to the power 100. Now, in generally one amino acid take 10 to the power minus 11 second. So, total time required for protein folding equals to 4 to the power 100 into 10 to the power minus 11 second. That will be something in that figure 10 to the power 41 year. That is more than the uh, time of the earth. Okay, but if a protein is taking only 100 amino acid protein containing protein is taking that much year, then how it's so random process, how it's so random that within so quite fraction of second the protein is synthesized, then folded, then exported to particular place, and it's functioning well. This is so random, right? That means that's actually not possible to fold a protein 5 into 10 to the power 41 year. Just assume 5 into 10 to the power 41 year is impossible. If one protein take that much time, then we will never survive. That's why it's called Levinthal paradox. That means some factors are responsible that will make the protein folding quite faster. What are those factors? Okay, we will see that. Now we will see the factors responsible for protein folding. First factor or enzyme, we can say better. Enzyme. First thing we will write protein disulfide isomerase PDI. Protein disulfide isomerase. What it does, guide disulfide bond formation. Disulfide bond formation. So suppose in earlier cases, suppose there is some protein, right? At 20 position one sulfide group is there, at 41 position some sulfide group is there, at 91 position sulfide group is there, at 120 something is there, 130 153 something is there, 170 something is there. But when the protein is folding, right, at that time here 20 is making bond with 120 suppose, or 120 or 91. How this is decided? That is by protein disulfide isomerase. It is making multiple disulfide isomerase, and then it's observing at which conformation the energy is minimum. We will see the energy concept. The energy is minimum because the minimum energy means the protein is stable. Another protein is PPI. Okay, that is uh, protein isomerase. So what is the PPI? Protein the PPI that actually help in cis trans isomerization. So if the proline in a side chain or in a stretch of amino acid is somehow converted from cis to trans, that can lead to uh, disrupting the structure of the protein. So PPI help. Suppose there is something, some strain is coming. So the PPI will help by converting cis to trans or trans to cis, depending on the situation of the protein. And the third thing is chaperone. Chaperone. This chaperone molecule are actually help is a heat shock resistant protein HSP. So this protein help the uh, protein or guide the protein to be properly so that the protein can fold properly. Okay, there are HSP 70, HSP 60, HSP 40 are there. So that chaperone actually work with expense of ATP. So these are ATP dependent process. The chaperone works with the expense of ATP only. Now uh, we have to see the thermodynamics behind the protein folding, right? So suppose some protein is like that. This is in unfolded condition. Then it is going through something intermediate. So that is unfolded. That is intermediate, right? And after that, the protein is properly folded. Now properly folded. Properly folded. How it is decided thermodynamically? The general equation for delta G is delta H minus T delta S. That is thermodynamic reaction. So generally, the most preferred, the most preferred uh, conformation at which energy is minimum. Energy is minimum. That means if you see the energy funnel, so the most stable protein lies here. So from here, suppose this is your native conformation, intermediate, your most stable protein is here. Your most stable protein, most stable conformation has least energy. Remember, most stable conformation or most stable protein has least energy. Now, you can do multiple things. Suppose the temperature has a role in protein folding. How you can see that? Generally, the delta G less than 0, that is a spontaneous reaction, right? Now delta G less than 0, that means delta H minus T delta S, that should be less than 0, right? So from here you can create multiple situations. So suppose delta S is, suppose delta H greater than 0, delta S less than 0. At that time delta G is always greater than 0. The reaction is always non-spontaneous. Non-spontaneous. But suppose delta H is greater than T delta S. At that time also the delta G greater than 0. At that time also non-spontaneous. Delta H less than T delta S. At that time delta G less than 0. At that time it will be spontaneous. Right? So the protein folding is a thermodynamic behavior. The temperature is deciding at which temperature your protein will be unfolded. Okay, now uh, so here it's little bit description of how chaperone help in protein folding. Suppose this is a protein, and if the protein is not properly folded, then what will happen? The side chain, everything is quite exposed and it's random. Then they will just attract each other. So suppose some charge residue are exposed, some hydrophobic residue are exposed, they are not buried properly, then it will form some random aggregate, right? So this is the protein, this is folding intermediate, that is the native protein that is properly folded. Okay, and what is the role of chaperone here? You can see the chaperone HSP 70, that is ATP dependent manner, it is consuming ATP, and then the protein is coming here and it's properly folding. Until and unless the protein is not properly folded, the cycle will be automatically continued. Okay, and in case of any misfolding, you have nothing to do. The cellular machinery should degrade the protein. There is no option if the protein is misfolded. But your cell of course will try to minimize this kind of misfolding. There are a lot of disease that is due to the protein misfolding, maximum the prion disease, Alzheimer, beta plaque, right? So here is also this is the ribosome and your protein is synthesized here. From here the protein intermediate things, then the native protein, right? The chaperone is helping in folding procedure. In case your protein is not folded, okay, now what will happen? That will aggregate, and that aggregation leads to different kind of disease and oligomer, amyloid fibrils. But actually chaperone also tries to prevent the aggregation and just to refold your protein again and again. And sometimes if refolding is not possible, just some ubiquitin mediated degradation. So what is ubiquitin mediated degradation? So suppose your protein is not properly folding, that means in our household, just think, our parents always tries to uh, rectify our mistake. So suppose in any case you are doing huge crime, okay, anyway this it is not possible, your rectification is not possible, you will never be corrected, then it's better that sometime the parents make the child uh, just okay, be in home, you don't have to go outside, you have to stay in home only, right? So that's the same quite condition. The in case the cell wants that okay, whatever protein I'm synthesizing, it should be properly folded, but in case something is not properly folded, okay, degrade, that is ubiquitin mediated degradation. So the protein will be ubiquitinated at lysine residue, and then it will act as a tag and it will be degraded. Okay, now what is the uh, factors that actually are responsible, factors that are responsible for uh, denaturation duration of protein? So the first thing comes in our mind, that is the temperature. How temperature thing do that? So by disrupting disrupting hydrogen bond, right? That's quite easy to break the hydrogen bond, and then if the hydrogen bond is broken, of course the secondary structure has gone. At that time your protein will never be at folded condition. Second thing is that suppose organic solvent organic solvent, if you apply some organic solvent, what will happen? The organic solvent is suppose OH, ethanol, or some COOH, something group is there, that group will make hydrogen bond. uh, this functional group this functional group will make hydrogen bond with uh, functional group of protein, functional group of peptide backbone, peptide backbone, right? And then also heavy metals, detergent, disrupt the hydrophobic interaction disrupted. Okay, and what else? Lot of things are possible. Okay, but in generally that are the most common factor that we use. So suppose in case of SDS page, if we're loading the sample, we use to give some heat to the sample just for denaturation. Okay, okay, so that's all about the protein folding, how protein fold, what's the role of chaperone, and what are actually the factor responsible for protein folding, and what's the factor responsible for protein denaturation, and a little bit of thermodynamics. Okay, so we'll stop here. And okay, sorry, so before that, before finishing the class, I would like to give you one brief information about collagen, keratin, and silk fibrin. So first one is collagen. What is collagen? Collagen is the triple, basically triple helix. So you can't say collagen as uh, alpha helix. So collagen is not alpha helix, but the pitch is here. You have to remember the pitch is 1.5.21 something. Okay, sorry, I might confused. So in case of collagen, the most important thing that three chains are interconnected to each other, and here the interchain hydrogen bond, interchain hydrogen bond is responsible for stability of uh, collagen. Another thing that collagen is most abundant among vertical protein, sorry, invertebrate protein. Second thing is that the general things that is required G, X, Y for collagen formation. G is glycine, X is proline, Y is maybe 4 hydroxy proline or 5 hydroxy lysine. Anything is possible. Now this hydroxylation of proline and lysine, that is done in PTM, post translational modification. Okay, and due to this hydroxylation of P or lysine, P and K need vitamin C as cofactor. So that's why vitamin C deficiency leads to scurvy, because the collagen is the gum. Yeah, like they form the gum, right? Now it's also copper dependent. So the hydroxylation is also copper dependent. Okay, now you can correlate like how different vitamin are responsible for the function and the formation of protein. That is, we say the protein is the bodybuilder. Okay, so if the vitamin C is not there, then collagen will not be formed, right? And the silk fibrin, okay, in another, and the silk fibrin, that is the anti-parallel beta strand, right? And another thing is uh, desmosine, desmosine that form elastin. That is the second most abundant protein after collagen. In desmosine, what happens here? Here, three modified uh, lysine plus one unmodified lysine. Okay, modified means they are already converted to CHO, and one unmodified lysine, they make desmosine, and desmosine is the main component of elastin. Okay, so that's all about the tertiary structure. Okay, and next time we will discuss the quaternary structure and of hemagglutinin and the cooperative binding. Okay, after that we will jump into the enzyme kinetics.